Hey, thank you, entire SRF community, for having me. It's my pleasure. Second time that I've attended this conference. The talk is not only about gene delivery, I'll also hint at gene editing a little bit throughout my presentation. When I was putting together my presentation for this meeting, I was trying to walk the fine line between catering towards the parents and the scientists in the community, because I think both are equally important. So I'm starting off with sharing some good news to actually show my appreciation of the parents or caregivers' support. What I'm going to show you today is that it is possible for us to deliver SYNGAP gene to the brain in a mouse model at least. I will share with you that we can perfectly send the cargo in these milk exosomes that I'm going to talk about to distinct tissues and distinct cell types where we want them to go. And I will point out, and that's also my conflict of interest disclosure, that we incorporated a company about 3 weeks ago, Minovaca, that is intended to accelerate the translation of what we discovered in the lab into patient care and in clinic. And our CEO, Staten Noel, is back in the room. If you want to talk with him later, Staten, we want to wave, yeah, back there, okay. Exosomes 101. So just envision the left cell maybe being a adipocyte, a fat cell. In these fat cells, hypothetical example, exosomes originate through endocytosis. They mature from early endosomes to late endosomes, end up in what's called MVB, multivesicular body, where they're loaded with a variety of cargos, RNA, DNA, proteins, lipids. And many of these exosomes are degraded in the donor cell, in lysosomes. Some are released in the cytosol, and very few are released in the extracellular space. Those released in the extracellular space travel to different tissue, for example, a brain cell, a recipient cell, and trigger all kinds of responses in this recipient cell, gene expression changes, metabolic changes, you name it. Where we came in was about 10 years ago, showing that exosomes do not exclusively originate in endogenous synthesis, but can also be absorbed from milk, from dietary sources. And these milk exosomes, as you will see, have possess qualities that make them ideal candidates for drug delivery. So in my lab, we engage in 2 streams of milk exosome, ME research. One of them is the delivery of therapeutics by using genetically and chemically modified milk exosomes. The other line of research is the importance of exosomes in milk for infant nutrition, particularly brain development. The questions that we ask are very, very similar in both streams of research, all about pharmacokinetics, delivery of cargos to certain tissues, effects of exosomes in the host. So my slides are also a little distorted, but I just walk you through what I have. So today's presentation obviously is about delivery of therapeutics, delivery of the SYNGAP gene. As I said, milk exosomes have lots of qualities conducive to the delivery of therapeutics. They can be loaded with a variety of therapeutic cargos, siRNA, plasmids, proteins, you name it. They're orally bioavailable, so it's a very non-invasive way to deliver therapeutics to patients. They're highly stable and resistant to the harsh conditions in the GI tract, and they protect the cargo as they're delivered orally. They can cross barriers, not just the intestinal mucosa. They can also cross the placenta. They can cross the blood-brain barrier. As far as we know, they are biologically safe. They don't elicit immune responses. They don't cause liver and kidney damage, and they don't disturb erythropoiesis. And they can be easily scaled up in production. So then there's a few publications I picked just to illustrate here. This is published research. Milk exosomes share with many other drug delivery platforms or vesicles a few limitations. First of all, they're rapidly eliminated by immune cells, by macrophages. They detect them as foreign and they just gobble them up like crazy. And like with many drug delivery vehicles, there's a lack of specificity. So they don't exclusively go to the tissue where you want them to go. They also end up in other tissues. And then the the third limitation is the extensive degradation in lysosomes. What I'm going to talk about today is the first two limitations, the elimination by macrophages and tissue homing, and how we overcome these limitations with our delivery platform. So this is a very important slide because it shows you the technology that we have developed. So what we are doing is developing programmable milk exosomes, PMEs, by using genetics approaches and click chemistry approaches. We generate these programmable milk exosomes in mammary cells because milk exosomes originate in the mammary gland, and because these mammary gland-derived milk exosomes show biodistribution and then bioavailability patterns just like milk exosomes. In this case, it was the HME, the human mammary cell line. So we genetically engineer these cells to load Cas9, a gene editor, into the programmable milk exosomes, but we do not, at this point, not include the guide RNA. Then there is 3 tetraspanins, 3 transmembrane proteins that you find in natural milk exosome, in any exosome, actually the CD9, the CD63, and the CD81. For our programmable milk exosomes, we focused on CD81 because unlike CD9 and CD63, CD81 is not glycosylated, which makes it much, much easier to work with them downstream when we modify and functionalize our programmable milk exosomes. And what you see here in the program, in the CD81 is we changed it a little bit. So we added in purple an extra transmembrane domain to take the Z terminus of CD81 to the outside of the exosome. And genetically, the C terminus has been fused with a his tag, histidine 6, which enables us to purify these exosomes from a very complex matrix, thereby decreasing the heterogeneity of the preparation, which will hopefully please regulatory agencies like FDA. In the CD81, we also substituted 3 unnatural amino acids for natural amino acids. So the unnatural amino acids are also called non-canonical amino acids, could be a phenylalanine azide, or an decoxamine, or an alkene. And these are on the outer surface of the exosomes. And now you have to envision this PME as a blank canvas. This is not functionalized in any way. So it can, the clinician or the user can actually functionalize toward the patient's needs. So we purify these PMEs by using nickel chromatography. We can clip off the histidine tag, this red tag, because it's a foreign substance. It's always a concern to the FDA. So we can remove the his tag after purification, and then we can functionalize this blank canvas toward the patients, to meet the patient's needs. So we can actually load the PME with guide RNA, which then would work with the Cas9, for example, to edit a mutation in the Syngap gene. We can also load the PME with a plasmid to deliver the Syngap gene to the brain. And we can use these non-canonical amino acids, the little circles in orange, blue, and pink, to attach short peptides that help us achieve our goals. One peptide could be used to attach a homing peptide, which will take the PME to the desired tissue. One non-canonical amino acid could be used to attach a don't eat me peptide, which prevents the macrophage uptake by these exosomes. And the third NCAA, this pink circle, could be used to attach what we call a retrofusion peptide or protein, which will decrease the degradation of PMEs in lysosomes. Click chemistry, I mean, 2 years ago, I did not understand how this works, but now I do. So this is how it goes. I mean, in click chemistry, look at CD81 here, we insert an artificial stop codon, TAG. Normal cells would stop translation of the mRNA at that point and would produce a truncated CD81, which is here at the bottom. If you equip the cells with the machinery necessary to read this stop codon as a codon that codes for an amino acid, that would mean you include an RS, RNA synthetase, tRNA pair, and feed the cells with a non-canonical amino acid, it will recognize the TAG codon as a codon that has a matching amino acid, a non-canonical amino acid, and it will express, the cell will express the full-length CD81 with the difference, or with the only one amino acid change. This is the non-canonical amino acid, which has been inserted in place of the natural amino acid in CD81, and the two amino acids look very, very similar and don't change the conformation of the CD81. So we tested the technology by transfecting cells with a fusion protein, CD81, and near-infrared fluorescent protein. And in the IRFP, we have the synthetic amber stop codon. And what we did, we equipped the cells with a machinery, RNA synthetase, tRNA, and fed the cells, or did not feed the cells with NCAA. Works like a charm. What you see far left, those are cells not transfected, so they cannot make the fluorescent protein, obviously. Second to the left is cells transfected with an IRFP plasmid, just to make sure the cells can actually make the IRFP, and you can see strong fluorescence. And now the two dishes to the right are the most interesting ones. They are cells equipped with a machinery to read the tag codon in this fusion protein as an actual amino acid encoding codon. Second from right is cells that have the machinery, but were not fed with the NCAA, the non-canonical amino acid, so they don't have the substrate. They cannot make the fluorescent protein. And far right, you can see the machinery is there, the cells are fed, and they do make the fusion protein. So this technology works. Quick reference to the Don't Eat Me peptide. When we decorated our PMEs with a classical Don't Eat Me peptide, CD47, the elimination of the PMEs from mouse plasma was drastically removed. But you see the CD47 positive, orange, yellow, plasma levels were much, much higher than the CD47 negative PMEs, because they were gobbled up, the negative ones were gobbled up by macrophages. This is not just a trivial observation, because it translates in a massive increase in the availability of these PMEs for tissue accumulation. So you see the CD47 accumulates much, much more positive exosomes, accumulate much, much more in the brain of mice compared to the CD47 negative exosomes. So in other words, it's the reduced elimination by macrophages makes more of the PMEs available for tissues. Next, we did a test run of our technology with IRFP plasmid loaded PMEs, delivered these reporter plasmids to the brain, to the mice by nasal spray. What you see, so the left panel, the couple of the pair of mice is the same like the pair on the right side. Left side is basically no densitometry analysis, but you can see the mouse that received the IRFP plasmid loaded PMEs actually express the plasmid in the brain. So the cargo is functional. So you can deliver functional cargoes by PMEs that are not degraded. And on the right side, you see the same pair of mice with densitometry analysis. And I apologize for the distorted images here. Of course, this is all not so relevant reporter genes. So next we moved on to testing SYNGAP gene delivery. And what you see here, and I boxed this for ease of following my presentation. So what you see boxed in yellow is a normal mice SYNGAP gene expression in the whole brain. In pink, you see the expression of the SYNGAP gene in a SYNGAP1 variant mouse. So definitely less expressed than in the wild type mouse. And in the far right, this turquoise bluish box, you can see our first attempt at delivering the SYNGAP plasmid to the brain. But what you can also tell, probably blue versus yellow, we way overshot in this first toss run. So there was way more SYNGAP gene expressed than is necessary in all likelihood. So we played around a little bit with the dosing. This was just the first test run. And now with these 3 replicates that you see here, 3 mice, or actually 3 sets of mice, compare, let's say, yellow to blue. And you can see we're getting really, really close to the expression of SYNGAP gene in wild-type mice. Homing. So now we asked the question, well, can we actually direct our PMEs to particular tissues? And we did so for multiple tissues to date, pancreas, heart, and hippocampus, as well as brain cancer cells. So what I'm going to show you is one example targeting the PMEs to the hippocampus. So we attach a homing peptide that is supposed to take the PMEs to the hippocampus. And what you can see here, I use my boxes again. Without homing peptides pink, with the homing peptide in blue, there's definitely an increase in the brain. But what's just as important is that you take away the PMEs from tissues where they're not supposed to go. For example, lung, there's definitely a massive decrease in the accumulation in the lung, and the same in the liver downstairs. And lung and liver are major sinks of exosomes, milk exosomes. A little higher resolution here. So now we're dissecting the same brain that you have seen to the left, looking at cortex, cerebellum, and hippocampus. And you can see the hippocampus peptide, homing peptide, really takes the PMEs to the hippocampus. Work in progress. So we're trying to look at the various cell types in the brain that actually take up our milk exosomes. We're not really happy with this slide. It's spatial profiling. I mean, looking at the cell types, but we really want to play around with this a little bit. Also, getting the dosing of the exosomes to the point where we want them to go. Currently, we are supported by the SRF. Thank you again. Doing some pretty boring work. I mean, boring for a scientist, but important when you approach regulatory agencies, because FDA will want to know, okay, what is the shelf life? What is the storage stability? How can you actually preserve these exosomes? Are they biologically safe? So this is kind of what we're doing right now. And I'll show you just one example here. It's about storage stability of milk exosomes. The dark yellow far left is just freshly prepared milk exosomes. The column in red is freeze-dried exosomes. You can see you lose actually almost 40 or more than 40% when you freeze-dry exosomes to have them in powdered form. And then the three bars to the right is milk exosomes that were lyophilized, I mean freeze-dried, in the presence of trehalose, which is the gold standard of preserving exosomes for pharmaceutical applications. Trehalose is an FDA-approved compound. If you like ice cream, you consume it probably every day because it's added to ice cream as an anti-ice crystal compound. So that works fine. So this is part of the things that we're currently doing. Conclusions would be milk exosomes have qualities conducive to the delivery of therapeutics, and the delivery of the SYNGAP gene is feasible. And this is the people and funding agencies that enable the work. I boxed the person who's actually all doing most of the work that I showed you today, Kamar Taban. So she's an outstanding postdoc in the lab. And with that, thank you.